It was right. Hi, everyone. Welcome to our Spatial Omics Seminar Series 6 again. And today we are very happy to have Dr. Anushka Dikshit from ACD Bio, part of the Bio-Techne brand. And as you guys heard in our discussion, they have a lot of different essays covering, including the RNAscope. And today we will have Dr. Dikshit from the company, and Dr. Dikshit received her degrees from Southern Illinois University School of Medicine, and after that she did a postdoc at Duke University before starting her career at the Bio-Techne, and currently she's the group lead of applications at the company. And without further ado, I'd like to turn over to Dr. Dikshit, please.
Thank you so much for the introduction, and I'm very happy to be here and talk to you guys about some of the new exciting assays that we have recently launched, and answer any of the questions you might have about this technology. So let's dive into today's presentation. And as the introduction said, today the topic is going to be focused on understanding single cell and spatial multi-omic profiling using our new version of the HiPlex assay, which allows for simultaneous detection of 12 and plus markers. So for today's agenda, I've kept it very broad, and followed by which I'll introduce the HiPlex assay as it was originally launched and talk about some of the historical applications, followed by which I would like to introduce the new version of our HiPlex assay, which is the HiPlex assay version 2 or V2, and then talk to you about some new applications that we have enabled with this new assay in detecting immune cells in tumor tissues. So let's start by doing a very quick overview of the RNAscope technology. A lot of you might already be familiar with this technology, but for those of you who haven't heard of us, we are an in situ hybridization platform primarily, and we were designed to overcome some of the tedious limitations of a traditional-ish assay, which prevents you from getting very specific signal, or a lot of the times these assays are marred by very high level of background and non-specific staining. So this technology was specifically designed to eliminate all of those aspects and give you clear signal detection of your target genes with a very seamless workflow and easy detection strategies. So this group is not new to understanding the importance of spatial biology, but I see over the last few years, a lot of researchers in the biomedical field are recognizing the importance of spatial detection of target genes or proteins. And this is because cellular organization helps us understand the fundamental function of a tissue or organ system. By visualizing the spatial context of a target gene or protein expression, we can understand how these different cells are communicating with each other. And this is important not only for cancer research or neuroscience, but for a wide range of research and application areas. These include developmental biology, inflammation research, and so forth. So overall, why is spatial transcriptomics important? Because it helps us elucidate cellular heterogeneity and study novel cell types that are identified by single cell sequencing, for example, in a spatial context. So overall, in the last few years, we've seen a lot of these cell atlases being developed for normal and pathological tissues. But once we have identified new cell types through these technologies, it's important to understand how do they sit within the tissue in a spatial context. And so a lot of the times before we are confident about these cell types within the tissue, it is important for us to visualize these cell types in a tissue context to better understand their function. And so a lot more of these single cell based technologies are now incorporating a spatial components to their platforms. And we are very happy to say that we have been in the spatial genomics market for almost a decade now. We're celebrating 10 years this year. And we have always realized the importance of spatial biology as we developed our technology and as we evolved our assays over the years. So let's go in a little bit more detail to understanding what this technology entails and how it stands apart from some of the other similar tools that are used by researchers. So the three key aspects of our RNA in situ hybridization platform include target probe designing, signal amplification, and detection. So ACT will design a specific target probes for your gene of interest, after which we have a proprietary signal amplification strategy that helps build very specific amplification trees, allowing detection with minimal background, providing very high signal to noise ratio. And followed by which we have detection systems that will allow you to visualize a single molecule of RNA with single cell resolution. And we enable this in a fluorescent and chromogenic platform. So overall, over the years, we have modified our assay to make it compatible with different sample types and for different RNA species, including mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, siRNAs, ASOs, point mutations, and so forth. So this incorporates a very large range of pathologically significant RNA species that can be visualized using this assay. Furthermore, this assay can be performed on a wide range of sample types, including FFPE tissues, fixed fresh frozen tissues, cultured cells, PBMCs, so forth. So because of the flexibility that we offer with this assay, we have been applied across different research areas and for various applications. So we have been used for neuroscience research to study developing embryos, in cancer and IO research, inflammation, for cell and gene therapy, and for infectious disease research. This last one has become very significant in the last couple of years, where we're very proud to say that we have several SARS-CoV-2 probes in our catalog that help advance COVID-19 research for some of the prominent vaccine developers like Moderna, for example. But overall, why is this technology so significant, and what are some of the advantages that we see with this platform compared to some of the other techniques that are out there? So because we are an RNA-based technology, we've been used, especially in neuroscience research, to look at a lot of the GPCR targets, which can be very challenging to study by IHC. Then we have been used to visualize secreted factors like cytokines and chemokines, and it allows us to identify the cellular source of these secreted factors and pinpoint which are the cellular subtypes that are secreting these important soluble factors. In addition, we have been used for validation of antibodies to understand their specificity, to visualize CDR3 sequences on TCRs to identify clonal population of T cells, then to visualize point mutations in cancer tissues. So it helps us identify these subclonal populations within big tumor samples that have these specific point mutations. Additionally, we have seen a lot of adaptability of this assay in cell and gene therapy because we can now visualize gene fusions, we can visualize copy number variations, and we can visualize integrated vectors using some of our other assays. And overall, just the flexibility of designing a probe against any target and any species has helped us penetrate different and unique research areas, which have struggled with some of the tools that they can use in the past. So The way we design our probes is extremely unique. I will go into the details of our amplification strategy in the next slide, but here I wanted to bring your attention to the way we design our probes. So here our probes are depicted as a Z molecule, and the way we design our probes, we have about 20 pairs of these probes that make up a single probe sequence that we use to target a target mRNA. So approximately, we span a range of 100 bases, such that pair of these probes are bound along these 1,000 bases of region in this fashion. So the probes that we provide are a pooled probe sequence. So here I'm going to talk to you about how these probes bind to your target and what really creates that high level of specificity and sensitivity afforded by this assay. So here we have depicted the probe as a Z molecule, and this is to highlight the different regions of the probe. So you have your target binding site region of the probe, which, as the name suggests, binds complementary to the target sequence of your interest. Then you have the preamplifier binding site, which forms the foundation for building the amplification tree. And then you have the linker region, which connects these two regions. So after you have your Z pairs bound to your target sequence, a preamplifier will bind onto a pair of these Z probes, followed by which you will have your amplifiers and your labeled probes. Now, these labeled probes can either be chromogenic or fluorescent based on the assay. So this ensures high level of sensitivity, and even the low expressing targets can be detected by this platform. Now, how do we ensure specificity? As I mentioned, this preamplifier binds only to a pair of Z probes. So if a single Z molecule non-specifically binds to another sequence that is not the intended target sequence, then the preamplifier will not bind onto the Z probe, and there won't be any amplification tree formed. So this ensures very high degree of sensitivity and specificity, giving you that high signal to noise ratio with a very low background. So in this slide, I have just put together all the different assays that fall under the RNAscope technology umbrella. Our parent assay is the RNAscope assay, which can be used for RNA targets that are over 300 nucleotides in length. And as I mentioned earlier, the probe design strategy for this assay is we design probes that are approximately spanning 1,000 bases, so approximately 20 pairs of the ZZ probes, because each Z will span approximately 20 bases of region. Currently, we have many different formats available in this assay, from different plexing levels to chromogenic versus fluorescent, manual versus automated, and so forth. Currently, this assay can go up to 48 plex in one of its formats. And next we have our BaseScope assay, which is designed for shorter sequences that are between 50 to 300 nucleotides in length. So these include your homologous sequences, if you want to span exon-exon junctions, or if you want to look at point mutations. The third assay, which we released last year, is our microRNAscope assay. As the name suggests, it is for detecting even smaller range of RNA targets between 17 to 50 nucleotides in length. And these are mainly your antisense oligos, siRNAs, microRNAs, and so forth. And finally, our latest DNAscope assay was designed to detect DNA targets. And so with this assay, we have really entered the realm of a new biomolecule, which is DNA. And currently we make this assay available as a duplex assay where you can visualize copy number variations, gene fusions, translocations, or integrated sequences in your genome. So in addition to all of this, we have also enabled co-detection of RNA and protein with 3 of our assays, that is the RNAscope, BaseScope, and microRNAscope assay, where you can visualize target RNA and a target protein simultaneously on the same section of the tissue. Now, this can be extremely valuable when you're looking at wanting to identify specific cell types using an antibody marker, and then visualize target gene expression using any of these platforms. So with that, for the purpose of today's presentation, as the title says, I'm going to focus on one of our assays within the RNAscope assay types. And this is our HiPlex assay. It is our latest multiplex fluorescent assay that enables detection of targets in both fixed frozen and FFPE tissues, and detection of up to 48 targets in fresh frozen tissues. So without further ado, let's jump into understanding how this assay works. So this assay was mainly developed so that researchers can detect 12 targets simultaneously on the same section of the tissue with single cell and spatial resolution. This became especially important to our neuroscience and developmental biology researchers, where you have sample limitation, where you only have a very small section of brain or small embryonic section to work with. So we wanted to maximize gene detection in a small section of tissue. And we released this assay with a very simple workflow. As you can see here, just like any other RNAscope assay, you will permeabilize your tissue section. You will hybridize all 12 of your target probes at the same time and develop the amplification tree on all the 12 probes for the 12 targets. And then you perform iterative detection of 4 targets or 3 targets at a time, based on which of our configurations you're using. So the way it works is you will add your fluorophores, you will develop the first 4 targets, cleave your fluorophores, add fluorophores to the next 4 set of targets, cleave, and so forth. So originally this was developed to detect 12 targets, but like I said, since then we have expanded the capability to allow detection of up to 48 targets. And this can be done by using a HiPlex up reagent that we introduced last year, where after you finish your 3 cycles of the 12-plex, you add your HiPlex up reagent, and this will denature the probes from your target sequence, and then you're free to add 12 new probes onto your target tissue. And then repeat this either twice to get 24-plex detection, 3 times for 36-plex detection, and 4 times for 48-plex detection. So internally, we have validated this assay and showed that it does not have any impact on tissue or RNA quality, because we use this chemical cleaving reagent for removing the fluorophores, which is extremely safe and does not damage the tissue or RNA, and conserves your tissue quality. So as I said, within this each round of detection, you can either do 3 cycles of 4 or 4 cycles of 3, depending on the capability of microscope you have, whether you have ability to detect 3 fluorophores or 4 fluorophores. And this assay was originally released in this format for fixed and fresh frozen samples. And in this slide, I just want to again go over how this iterative modality of detection would work, where, as I mentioned, you would hybridize 12 probes at the same time. Now, each of these 12 probes have these unique tails, 1 to 12, and this unique tails will help develop specific signal for each of these probes. So what happens is, after you hybridize and develop the amplification trees for all 12 targets, you will introduce your 4 fluorophores for the first 4 targets. After you detect your add your fluorophores, you will image the first 4 targets, followed by which you will add the cleaving reagent and cleave the fluorophores of the first 4 targets. Then you will add the fluorophores onto your next 4 targets, image the next 4 targets, and then proceed to do your last cycle of adding fluorophores to your last 4 targets and then imaging the last 4. So this is how the iterative modality of detection works for this HiPlex assay. Ultimately, we realized one of the biggest applications of this assay is for researchers performing single cell sequencing or to develop new cell atlases or profile tumor tissues, brain tissues, embryonic tissues to identify new cell types. But after they have done that, uh, they are very keen on visualizing these unique cell types at the tissue level. And that's where we provide our HiPlex and other multiplex assays for orthogonal validation of the single cell sequencing tools. So after you've performed your unbiased gene expression analysis by single cell sequencing, you identify those key markers that are differentially expressed or are novel cell type markers, and then you validate their expression with a spatial context using this HiPlex assay. So this ultimately enables developing new cell atlases, identifying and visualizing new cell subtypes to confirm target cells for new therapeutic interventions, and so forth. So we wanted to see how well this would actually apply for a neuroscience project, since this assay was released for frozen tissues. And to do that, we looked at this paper that came out in 2016 that profiled the mouse striatum for identifying novel neuronal subtypes within the mouse striatum. And so this particular paper showed how they profiled the striatum by doing cell sorting, followed by single cell sequencing, and identifying novel cell types within the mouse striatum. So what we wanted to do was look at this research paper, identify the different targets that they have discovered using single cell sequencing, and then use our HiPlex assay platform to visualize these novel neuronal populations in a tissue context. So to do that, we looked at the paper and identified the different targets that they had discovered for D1 specific medium spiny neurons or D2 specific medium spiny neurons, and then combine that with a couple of targets that are expressed in both D1 and D2 positive neurons, and then use our 12-plex assay to visualize these neurons and their subpopulations in the tissue. So here you can see that we have visualized a set of targets specific for the D1 population of neurons. And this allows you to see how well this assay works to give you that single cell and single molecule resolution, where now we can visualize these targets with spatial context and even identify the subpopulations of the D1 neurons, where you have subpopulations that express a set of targets versus this set of population that express a different set of targets. So this really allowed us to visualize subpopulations of the medium spiny neurons and then identify the subpopulations that were already discovered in the paper, but visualize them with the spatial context using the HiPlex assay. So similarly, for the D2 population of neurons, we took a set of targets, which were specific gene signatures for those neurons, and then identified subpopulations within this D2 population of neurons. Again, highlighting how well we are able to look at a single molecule of RNA at that single cell level. So overall, what we realized is this assay is able to provide that spatial context which enables you to visualize your heterogeneous population of neurons in the same field of view and understand how are they distributed spatially within a section of the striatum here. So we just used this GIF and using our image registration software, created this beautiful image. Speaking of registration, it's all well and good creating a beautiful image and allowing 12-plex detection, but how do you analyze this data? That is the big question. And so we released the image registration software along with our HiPlex assay, which allows you to overlay your 3 or 4 imaging cycles and create this single image where you can now toggle on and off between all 12 targets and visualize their expression in the same image. So this really allows you to study individual subpopulation of these neurons and study their spatial organization. What was important was in this paper, in this group, they also identified that although majority of the neurons were exclusively DRD1 positive or DRD2 positive, there were some hybrid neurons that did express both DRD1 and DRD2 expression. And this was a very novel find from their single cell analysis by this team. And what was really interesting was using the HiPlex assay, we were able to confirm that and visualize those hybrid population of neurons in these tissue sections, which was really fulfilling to see that we're able to show what was already discovered at that spatial and single cell resolution. So this assay has really taken off. We have some really cool publications, mainly in neuroscience research, some in developing cell atlases with frozen tissues. But the big question we kept getting asked over the years was, can this assay actually be used for FFPE tissues? And at that time, we released this assay only for fixed frozen tissues, but our R&D team was hard at work to make this assay compatible with FFPE tissues. And then this year, we are able to say that, yes, you can use this assay for FFPE tissues because we launched our HiPlex version 2 assay, which is now compatible with FFPE fixed and fresh frozen tissues. So we were very happy to release this assay because it helps our researchers to apply this not to a single sample type, but to one of the most popular sample types, especially when it comes to disease pathology. So how does this new HiPlex version 2 assay really work? We have not modified this assay significantly, especially with respect to the core technology and the workflow. It is still very similar to our HiPlex version 1 assay. The workflow is identical. We have only introduced a couple of more reagents to the workflow to enable optimum detection of signals on FFPE tissues. And this was a very tall order, especially considering a lot of the autofluorescence issues that we experience with FFPE, and not just for ISH or in situ hybridization, but for IHC as well. And so our R&D scientists work very hard to develop a reagent that helped us combat autofluorescence and provide very specific signal detection for our RNA targets. The workflow, as you can see, remains exactly the same, where you permeabilize your tissue, you hybridize your probes, you develop your amplification trees for your probes, and then you perform that iterative modality of detection for 4 targets at a time. But now we have introduced this FFPE reagent, which you add as a part of the workflow to enable optimum detection of your target signals. So in the next slide, I want to talk to you about where this reagent goes and how it impacts the workflow overall. So we can see that this is your standard workflow where you add your probes, you develop your amplification trees, and then right before you add your fluorophores for detection, you add your HiPlex or your FFPE reagent for a 30-minute incubation time, and then develop your first round of signals. Now, this is what you absolutely need when you're working with FFPE reagents. But in some cases, when you're working with certain highly autofluorescent tissues that are stubborn, that have a lot of blood vessels, etc., you have the option of using this FFPE reagent more than one time as a part of your workflow. So you can add it a second time, again, between your first and second rounds, or rather right before you develop your second round of probes, and add it even a third time before developing your third round of probes. So this way you can ensure for targets that, or for samples that have very high autofluorescence, you're not seeing any non-specific background, or your true signal is not getting masked by autofluorescence. And in the next couple of slides, I want to show you how well this assay works for highly autofluorescent tissues. So this is an example of a tumor tissue where we have treated this with our HiPlex, standard HiPlex assay or standard multiplex assay without our FFPE reagent. And you can see that there is a high degree of autofluorescence to the point where you don't really see any real signal. And these are your, obviously your high expressing targets are positive control probes. But using our FFPE reagent, we were able to show that this ovarian cancer tumor now looks really good, and we can visualize your targets that are your PPIB, UBC, and polar 2A high expressing targets at that single cell, single molecule resolution. So it is not only eliminating your autofluorescence, but it is not masking your real signal. So this really helped us build confidence in how well this assay works for FFP tissue. We tested this not only across tumors, but different sample types as well. But ultimately, the question we were getting was, how does this compare to the original HiPlex assay, which was our version 1? So we compared the performance between HiPlex version 1 and version 2 on some of these frozen brain sample tissues. And as you can see, we see that the signals were very comparable between the two. We're not really seeing any performance differences. We are not compromising on signal quality by introducing this FFPE reagent. And more importantly, the new version 2 assay, while compatible and optimal for FFPE, is still very much compatible with frozen tissue samples. So this was very important to us to show our existing HiPlex version 1 customers that we have not compromised on signal quality. And next, we wanted to show the comparable signal detection with our new HiPlex assay to that of our current FFPE assay for multiplexing, which is our RNAscope multiplex fluorescent assay that allows you to do 4 targets at the same time on FFPE tissues. This is our go-to assay for multiplexing for FFPE tissues currently and works extremely well with, you can see the strong signal detection. But when we FFPE, we could show that the expression levels were comparable, the signals were just as crisp, and we can still go down to that single cell resolution. So again, we are not compromising on detection on FFPE tissues compared to our current on-market assay for FFPE multiplexing. But there's a slight advantage with the new assay where now we're not limited to just doing the 4 targets. We can now do up to 12 targets on FFPE tissues on the same section. So this was a very big advancement for us, considering how important FFPE tissues and how relevant they are for new therapeutic development, identification of new biomarkers, and so forth. So in the next few slides, I want to talk to you guys about the different applications now enabled by this HiPlex version 2 assay. So as I mentioned, validation of single cell sequencing remains one of our biggest applications because it allows you to validate a nice group of 12 targets on the same section of the tissue. But in addition to that, we are also applicable to tissue profiling of your heterogeneous tissues like brain, tumors, or developing embryos, where even if you're not performing single cell sequencing and you're wanting to explore, for example, a signaling pathway or compare treated versus untreated tissues for a set of biomarkers, you can use this assay for that level of gene expression profiling, where it will enable detection of your targets with that spatial resolution. And that makes it applicable to all of these research areas, where in neuroscience, you can profile different regions within the brain and identify novel cell populations and visualize these novel cell populations. For immunology or inflammation research, you can identify new immune cell subtypes. You can identify their activation states by looking at cytokine and chemokine expression, and so forth. For cancer and IO research, as you can imagine, you can study the gene expression in the dynamic tumor microenvironment, identify gene markers or gene signatures specific to certain cell types within the tumor microenvironment, and so forth. For developing embryos or in developmental biology, stem cell research, you can study the transitional cells, which are your cells that showcase expression of multiple different cell types. And these transitions, and they are very important in understanding initiations of certain childhood cancers. In addition, you can study disease pathology. We have a lot of papers showing how well this technology works in fibrotic tissues, in tissues with high level of collagen, and so forth. And some of the other applications, as I mentioned, is to develop and validate cell atlases and compare disease and normal states of tissues. But for the purpose of today's presentation, I want to focus on the cancer immuno-oncology research area and show you an example of how we have used this RNAscope version 2 assay to profile the tumor microenvironment. So why is it so important to profile tumor microenvironment in cancer? Because it helps us characterize these tumors and understand precise tumor protein expression. It helps us study the expression of your targets that might be important biomarkers or therapeutic targets, and define their expression patterns in different stages of tumors or with different patient populations. Additionally, as I mentioned, it is important to study not only infiltration of immune cells, but understand their phenotype and their activation states. And this becomes especially important when you're developing immunotherapies to understand their efficacy. And finally, it can provide information about initiation, progression, and metastases by identifying specific gene signatures on your target cells. So all of these are important considerations when you're developing new therapies for cancer research. And so, as I mentioned, I want to show you one example internally of how we profiled tumor tissues by using a set of markers that are implicated in cancer. So the workflow was very simple. We used a tumor TMA and we profiled using our new HiPlex version 2 assay, where we probed using all 12 targets and then developed 4 signals at a time, and then imaged and used our image registration software to combine these images, and then later did image analysis using some other quantification software. So let's look at some of the images that we got by using the HiPlex version 2 assay on the tumor TMA. So we have here expression for all of these relevant targets. You have CD3, CD8 expression, interferon gamma, PD1, CD68, CD163, arginase 1, CCL22, CXCL10, and so forth. So these are a nice combination of your immune cell markers, cytokines, macrophage T cell markers, and some hypoxia markers as well. And here we are looking at expression of all of these 12 targets in these 4 different tumor types. But obviously, although these images look extremely pretty, for us to actually get some biological understanding of what's happening in the tissue, we need to study individual expression of these targets. And that's where our image registration software comes into picture. So along with the version 2 of the assay, we have also released a version 2, a new and improved version of the image registration software. And some of the new features of this registration software are a larger display area. We have an auto color assignment. So when you overlay all your images, it automatically assigns different colors to each target, which obviously you can go back and change. Then we have zooming capabilities where after you combine, you can zoom in or out to get a deeper view of your image. Next, we have introduced a very important new feature as a part of this new version 2 registration software, which is background removal. So this particular feature is extremely important because it allows you to take a blank image of your tissue section in between your HiPlex assay and use that to subtract any background of your images after you perform your HiPlex assay. So this way you don't have any non-specific background signal and you get clear, crisp images with true signal. And so this was a very important feature that a lot of our customers are very excited to start using. So with this new image registration software, here's an example where we show that this was a section with very high population of blood cells infiltrated here. And even with the HiPlex FFPE reagent where we reduce the autofluorescence, we can't really get rid of these really bright signal coming from red blood cells, for example, in certain cases. So in that case, we can take a blank image where you have your only autofluorescence image, and then you can use that to subtract from your true signal, true image with your probe signal. And with the background removal feature of the software, you can only now see your true signal from your RNAscope probes. So it's a very important feature which allows you to study your real signal and also helps later in quantification of that signal. So here's a demonstration of how the image looks when you overlay all 12 targets simultaneously. Now, once you've seen all of these targets individually, you can tell that all of these are very specifically expressed in their target type, but you can't really tell which target is expressing in which set of immune cell subtypes. So then using the image registration software, you can toggle between targets. Here I have turned off expression of all the other targets expressed except for CD3, CD8, interferon gamma, PD1. So it helps me identify, okay, from all of these expressions that I'm seeing here, where are the T cells located within this tumor? And helps you study the spatial organization of these different subtypes. Now, here is again an example of a set of markers specific for macrophages, and you can toggle on and off between targets to see out of these macrophages, you potentially have CXCL10 being expressed by only that set of cells expressed on the top left corner. So you can make some really nice conclusions and study the spatial organization of these infiltrating immune cells using this assay. Furthermore, because we allow for the single cell, single molecule resolution, you can focus on a specific region of interest within your tumor sample, go down to that single cell resolution and identify different subpopulation of your immune cells. So here you have CD8 positive cytotoxic T cells that have all of these markers expressed. Then we also found a set of T cells that were CD8 negative, but CD3 interferon gamma PD1 positive. Then we found a set of cells that were interferon gamma negative, but were positive for some of the other CD8 markers. So you can make conclusions about what are the activation state of these T cells, for example. Similarly, here we have looked at some of the popular macrophage markers, CD8, CD168, arginase 1, CCL22, and you can make some comments about which are the cells that are expressing CCL22 versus cells negative for CCL22. And if you have a set of markers co-localized, then you can make conclusions about whether it's an M1 or an M2 macrophage based on the expression levels. So you can make these type of conclusions visually. You can get a qualitative analysis of how your infiltrated immune cells are distributed. But ultimately, if you want to do some quantification analysis, we have that ability as well. So here's an example. We have in the same study, we used some of the probes specific for studying hypoxia, like VEGF or HIF1 alpha, and we coded quantification using this was using, I believe, QPath, which is an open source software, and we were able to count a number of copies for each of these target per cell. Now, this is just the analysis we wanted for this, this type of targets, but you may not always want the copy numbers for each of the targets. What if you only want to understand positive or negative expression of a marker? We enable that type of analysis as well. And using softwares like, for example, Halo, provided by Indica Labs, you can do that high level of quantification of your RNAscope signal. So here's just a quick workflow of how we went about doing that quantification. So we selected an ROI, and using the halo analysis, we defined certain areas. We defined fish scoring criteria. We made sure that we are detecting the DAPI signals correctly so that we identify the true cells and not any background, and then perform quantification by using several modules within Halo specific for RNAscope. And with that type of analysis, we can get to quantify cells with different phenotypes. For example, here we have CD3, CD8 positive cells. And out of the 25% T cells, which are the total number of T cells within this ROI, you have about 12 or 13% that are CD8 positive. You have about 8% that are CD8, CD3, and interferon gamma positive, and so forth. So depending on what your ultimate hypothesis is and what your goal is for this assay, you can either count copy numbers for each of the targets, or you can just define phenotypes and percent positivity of those phenotypes within a given cell population. And we are working on developing an app note where we will detail how to best quantify RNAscope signal using Halo. But overall, I believe today I've shown you how well our 12-plex HiPlex version 2 assay works on FFPE tissues. We have eliminated the autofluorescence that is often seen with these tissues, making it difficult to see real signal. In this example that I showed you, I've shown you how well you can discern between immune cells and immune cell subpopulations and study expression of activation and inhibition markers. With our new image registration software, we have included the background subtraction option, which helps you create even better, crisper images while maintaining the true signal. So we are very proud of this assay since we have enabled this on FFPE tissues, and not just made compatible, but optimized for FFPE tissues. And we hope that this will allow advancement of research areas like oncology, immuno-oncology, inflammation, and so forth. So with that, I'll take any questions. Thank you.
Yeah, thank you very much. This is a terrific presentation. And also thanks so much to all your colleagues that really answered the question in the chat box in real time.
That's excellent.
Yeah, that's excellent. So it looks like most of the questions in the chat box have already been answered, but you can go ahead and pick the ones you think you want to further provide more details. But the last question, maybe you can I can rate for you, or if you can ask in the chat box.
Yeah, I was just saying, I think most of the questions have been answered.
Yeah. Yeah, and the last question from Ameka Unwell. So the question is, I would like to echo a previous question, which has now been answered. So what fraction of target molecules are detected? And along the same line, how much sensitivity loss during the different staining cycles, and do you have any data or evidence to prove, to support that?
Yes, and today I presented the high-level data, of course, but as a requirement of our product development, we have feasibility studies where we test different rounds and then check for any loss of signal, any impact on tissue quality, and things like that. And so all of this assay has gone through all of that rigorous testing, and we have showed that between rounds, we don't see any impact on signal quality. We don't see any loss of signal, even if we do same targets. So we have taken care of all of that and made sure that our different rounds or iterative cycles of detection are not impacting signal quality. So ideally, you shouldn't see any signal loss. And I think there's a sensitivity question. So in our technology paper, we have seen that we can actually goal cell molecule. So in theory, we detect each and every molecule of RNA, but sensitivity can be impacted by a lot of other factors, sample type, your workflow, technical, considerations. So considering all of that, we have still seen very high level of sensitivity, upward of 90%.
Okay, thanks a lot. But on the other hand, I think people in the sequencing space, the single cell sequencing space, they never got a 90% sensitivity, right? The RNA capture efficiency is like a 20%, but it's still very meaningful. So in this case, I wonder if you guys, it's probably not just for RNAscope, but for all other kind of in situ kind of facial approaches, whether or not any kind of normalization, once you get the data, quantify any kind of normalization and a batch correction also helpful for this type of in situ gene expression data analysis.
Sorry, Ron, I completely lost you for like 5 seconds there. Do you mind repeating the question? Oh,
okay. Yeah, my question is, I think in a single RNA sequencing data, we never got like 90% of sensitivity. Oftentimes we can detect only like a 20% or 25% of the transcripts, but the data is still very, very useful, very meaningful. And largely because you can do normalization, you can even do batch correction. So I wonder if the same thing, the same approach can be applied to the in situ data analysis. This is not just for RNAscope, but probably broader for all other in situ gene expression analysis technologies as well. I just wonder whether anyone thought about this.
Yeah, no, that's actually a great point. And with sequencing and this, I mean, since you're the expert, I'll ask you the question of, do you think it does bias for, some of the high expressors, versus some of rather the lower expressing targets? And I think that bias is sort of eliminated in this platform where you get the true expression of or whatever targets you're profiling tissue.
Yeah, and so along those lines also, when you compare the assays, the rigorous comparisons of like fluorescence version 1, 2, HiPlex, right? Do you guys have any like quantification framework? Because when you mention, oh, this works, this is better, and I'm curious, like if there is any like a plot that shows like number of transcripts for the same sample, right? And these kind of things, because when you do these kind of assessments, I think it will help scientists to find their own appropriate assays, right?
Correct. Show the quantification for the comparison, right? Yep, that will be very helpful.
Yeah. So I think a good example is from Caltech, another professor called Niles Pierce. If you look at some of his papers, he's very rigorous in some of the analytics. I remember actually spending days on just generating one plot on the accuracy of some of these assays. So I would highly suggest, looking at maybe some similar methods for RNAscope, because they're beautiful images, but quantitatively they will be really cool and helpful.
No, and that's a great point. And to your point, we do have a few papers where, researchers on their end have done comparative analysis between different assays. So for example, we have a couple of papers where they have done quantification with RNAscope, and then they have done PCR for the same targets. And then they've shown the comparison of, number of transcripts in that way. And that we found some really nice papers showing nice comparable expression between assays. So that's really also helped us build confidence that we are actually capturing what most of the other gold standard technologies are able to capture.
Exactly. So I think when we also did this with like even fish, the low copy number detection versus high copy number detection, the error bars, they get a lot bigger in the low copy, right?
Right.
Versus the high copy. And that's probably true in RNA sequencing too, I guess, Ron. So like these kind of things would be interesting to see also for RNAscope, like, what are the detection advantages or disadvantages? Because it will kind of affect our scientific questions as well, right? When we look at, for instance, the CD8, CD3 copies, I'm not sure how many copies that is per like cell in immune cells, but to identify an immune cell as CD3, what's our threshold, right? If there's a non-specific binding of like 3 or something, then will we accept it as immune cell, or is it some other cell that we will not accept it? I think in the field, I think you're wrong. Also, even in the imaging data, there are these challenges of like, where do you really set your threshold? Because there's still some non-specific things going on in the tissues and samples.
Yes,
Yeah.
and yeah, for sure. And sorry, I moved because my connection was getting bad there. We have, so Halo in itself has a lot of tools and modules where you can kind of train the software to eliminate some of the non-specific staining, or make sure that you're only capturing the real copies. So obviously, and that is something that, you know, as an RNAscope user, you have to learn as more and more analyses you do. So I think after doing it for a little while, you become more proficient in, you know, making sure you're capturing the real signal and getting the right quantification with those platforms like Halo. So I know they do have some nice modules, but yep, we need to make sure that, you know, our users are correctly trained on how to use those softwares and how to best sort of like best practices for RNAscope staining detection. And so that's why we thought for this assay, because it's a 12-plex assay, and, you know, the higher you plex with in situ, the more complicated data analysis and quantification becomes. So we really want to kind of release that application note where internally we are going to work on the data analysis and kind of put forth like a demonstration and best practices of how to accurately quantify.
Exactly. And one more thing that I remember is the TCR BCR detection will be actually very cool nowadays. And when I talk to actually people about in situ, they immediately ask, "Can you guys do it? You know, TCR BCR?" What do you guys think? Like, where is the future?
We have a lot of papers on TCR detection or CDR3 detection, and our BaseScope assay is specifically meant for that highly homologous target detection. So it can capture those you know, small regions which will differentiate, for example, clonal populations, and target those. So I'll be very happy to send you some of those papers where they have used CDR3 probes and such.
Mm-hmm.
Oh, you are able to detect the different TCR clones? Oh,
yes.
Wow, that's pretty impressive.
Yeah, it would be great actually. We can take a look because, you know, upon these infections, right? How many of these variants do we get? Would be great to know. And, you know, Mark Davis, for instance, at Stanford, they've been interested in this using sequencing. And they discovered like specific patient, you know, things, specific ones. And and if you can do this in situ, right, in specific target like tumors, right, and that will be actually fantastic. And we are happy to look at your papers, yes. Mm-hmm.
But in this case, you have to know the target TCR sequence. Otherwise, I think each naive T cell has different TCR. It's probably unlikely you can look at all different T cells.
Yeah, you
exactly.
probably need barcoding, I guess, right? Because you need like probably how many differences?
Yeah, you have to sequence basically. You have to
Correct.
do discovery.
Yeah, image-based sequencing. Yeah, exactly.
Yeah. Also, I think I have a question that's really kind of scientific. This really scientific question. Also inspired by what Ahmet was asking. Sort of the noise, if you see whether or not it's non-specific or some sort of background noise versus the real cell-to-cell variability, right? Because people know from the low copy number genes, the sort of the gene expression variability actually, or the noise actually dictate what the cell's gonna do to choose their kind of fate decision. So I wonder if you can kind of quantitatively differentiate so the level of noise versus the true stochastic gene expression variability. Maybe a very cool experiment to do is you can do 12-plex, right? But every 3 maybe in the first three probes or three genes you look at. The second round, you look at the same one. The third round, you look at the same thing. Then you can quantify the technical variability, and then you know how much variability you see across different cells is above that level, and then that's real. That actually tells you interesting biology.
Correct. If you see them at the same expression level over and over again, in the different rounds. And I'll just like to mention that you're talking about like high noise with low expressors, because of the way we design our probes, right? And the amplification tree, the threshold that we have for any level of signal detection. So you can see that there's variability in dot size. The smaller the dot means the number of Z probes bound is different than for the larger dots, where you have more Z probes bound, right? So the threshold is about 3 pairs. So for any kind of signal to appear, at least 3 pairs have to be bound to the transcript. So that sometimes also enables us to visualize, say, degraded RNA transcripts, which can be, tricky to pick up by some of the other assays, and something that's seen a lot in archival FFPE tissues. So, that would be a bit challenging how to separate out noise in that case.
Okay. Yeah, I don't know if anyone want to unmute yourself and ask a question. I see Steve.
We just keep the discussion going. And in the meantime, as we wait, maybe, is there any amplification variability that you guys observe? Like there's a distribution. I mean, those Z probes, like there's some stochasticity of binding, right? Still.
You mean for different target transcripts, the probes for different transcripts? So we do have our probe design team where we go like through what we call feasibility of probe design. So they have a certain list of criteria that have to be met before the probes can be designed for the target. So if there's high GC content or things like that, they have ways to work around that on the target sequence, given that it's long enough, so that it meets all the parameters. So we try to keep everything very uniform for all of our target probes.
So it's a drug delta function. There's no variability.
Not in theory.
So I mean, based on like our lab's experience, for instance, there's always this range of difference due to the RNA quality, due to the assay sample, and many other things, right?
Correct.
So that I think we observe, and that's why like amplification, when you do it, I think we always have this thing that, okay, maybe 10-20%, we have this room to kind of think about that this could be more or less, right? So
I was talking purely from a probe design perspective, like that, like all the parameters we have to meet for the probe design are pretty uniform for each target, but then there's so many other variables, like you said, sample quality, the protocol work, technique, and those can definitely impact. But I mean, I have done the homebrew-ish assay during my PhD, where I cloned the probes and did everything myself, and that like required RNase free environment, and I had to wipe down the incubator from top to bottom, and so all of that, I think we've really come above all of that with this assay, where you just need a simple lab with no stringent conditions, really.
Great. Any other question from the audience?
Yeah, feel free to unmute and ask if you are able to.
And if not, then let's thank Dr. Dikshit for this fantastic presentation again. And I'll use this reaction clapping as well as physical. And this was great to see the most recent developments from a very famous company. And before we wrap up today, then next week we will have Dr. Alistair Boetiger from Stanford University, a Murphish pioneer, I would say, and he's also on this call today. And please remember to dial in next week. And he's going to talk about genome origami differentiating cells by bending the blueprints. And with this, thank you guys again, everyone, for participating. And that was
Thank you for allowing us to present. Very happy to present to this audience and for the opportunity.
Thank you, Dr. Dikshit. And also the recording will be available on the website, spatialomics.net, as well as our YouTube channel. Please feel free to check out. And thank you, everyone. And please have a nice weekend and hope to see you guys next week.
Thank you.
Yeah. Thank you, everyone. Have a good weekend.